So you're ready to upload your first chemistry lab report in Canvas, and you need a little help about how to get started. So let's take a look. The first thing you need to know is, where do you find your lab report in Canvas? And you just go to the module that describes that experiment. So it's the place where you would have downloaded or just looked at the procedure for the experiment and taken the safety quiz and so on. And down at the bottom, there'll be a place where you can enter a Canvas quiz, actually, and that Canvas quiz is how we enter our lab reports, okay? So you're gonna click on there. And when you do, you'll go into a series of questions. So it looks like you're taking a quiz, and it's really you're entering individual pieces of your lab report. So the very first question is to enter your unknown number. So that would have been the number that was on your vial or your bag of vials that you started with. You're going to be asked to upload an IR spectrum. So, when you print out your IR spectrum, it's very good practice to have taken a picture of it right away, just in case you lose it, okay? Or you might take another picture after you've worked on your IR spectrum a little while and you've gotten some things labeled on it, and then you can just upload that here. And then it asks you to fill in a table, a data table, that you've learned in other presentations how to fill in data tables for different kinds of spectroscopy. Okay, so all that goes in here. I should say that after you filled something in here, it gets saved, so you can navigate away and it'll still be there. You don't have to do it all at once. And you don't want to hit submit until you're finished with everything. So once you hit submit, it thinks you're done with the entire report, even if you've left something blank. So here, for instance, is a data table. This is not IR spectroscopy. This is proton NMR spectroscopy. But I'm just showing it to you because I wanted to point out something that you may have seen before, but just in case. This is an NMR table that contains actually more than one compound. And I've got a line running through the middle of that table to signal that I'm organizing this table so that one compound is above that line and another compound is below that line. This often makes things much easier for you to organize if you do it this way. And the other thing that I'm going to point out here is I've got this superscript number up here in this part, and that's called a reference callout. That means I used some sort of reference source to assign those part structures, and later on I'm going to tell you what that source was, okay? But putting that call out there is a signal to the reader that you were using some sort of reference for that, and somebody who's grading this is going to be looking for things like that, okay? So now one of the reasons people will lay different compounds out into different parts of this table, by the way, is because then when they're looking at the integrals for this lower compound, they only have to look at the other things that are in that part of the table and worry about the ratios between the peak at 3.22 and the peak at 1.19, for instance. Or when they're looking at the upper part of the table, they only have to worry about the ratios between the integrals of those first three peaks, and they can ignore the other two. Otherwise, it can be a little confusing to see these peaks that don't belong as you go through. But at the end, if you want to figure out the ratio of those two compounds, if you want to work out the percent composition, it becomes fairly straightforward, just comparing a line in the top part of the table to a line in the bottom part of the table, and what is the ratio between those. That'll lead to your percent composition. But that's another presentation altogether. So there are a lot of data resources for you, and on the front page of Canvas, you can click on a tab that says Data Analysis Resources or Data Resources, And it'll lead you to a list of different things, such as how to use the UV spectrometer or how to download the NMR software and so on. Okay, so that's useful to know about when you're writing your report. One thing that students often use is there's an attached article that has some useful tables in it. That's the solvent impurity article, third from the bottom there. So I'm just going to look at that quickly in the next slide here. Here's one of those tables, and I want you to look at this column that says if you're using chloroform for your proton NMR spectrum. This is the chloroform column. And if you have any of these solvents left in your sample, where will they show up? Well, if you used water at any point in the experiment, it might show up around 1.5, 1.6 parts per million. If there was ever any acetone used, we often wash our glassware with acetone, it could show up at 2.1 parts per million. Now, because we see those peaks doesn't mean for sure that's what it is. It just means that's one of the possibilities of what you might have in there. Okay, tert-butyl methyl ether, I'm going to point out because it actually contains 2 peaks. There's one at 1.2 and one at 3.2. So 2 different peaks in that spectrum. And if you see both of those, that might mean you've got some tert-butyl methyl ether left in your sample. Okay, so consulting this table before you start worrying about what else is in your NMR spectrum can save you a lot of work, because it can eliminate a lot of the extra peaks that maybe don't actually belong to your compound after all. So once you've gone through and you've filled in your data tables and you've got some idea of what you're working with, then it might be time to go and confirm what you've got. This often makes students feel more confident about what they're saying. And it's also a requirement for 1 point, a relatively small amount of points, but that you've gone out and you've tried to get a reference spectrum, compare it to yours, just to make sure you are on the right track. I would not do this as a first step when you're writing your report. Students they waste a lot of time scrolling through these resources, looking at spectrum after spectrum after spectrum, trying to compare their whole sample to each of these spectra in the literature. It's too time consuming. You can narrow it down to a couple of compounds or even one compound and just confirm by looking at one example whether you've got the right thing or not. So I would save this to the end, okay? No matter what some other student tells you, listen to me, not them. Save this to the end. So where do you find this stuff? One source is SDBS. You can find examples of spectra to compare to yours. And this is from the National Institute of Science and Technology of Japan. You scroll down here and there's a disclaimer button. Disclaimer button you've got to click on before it lets you in. And once it lets you in, it's got a search page. And so, for instance, if you go through the compound names in the lab manual and you search for the name of the compound that you think corresponds to your structure and type it in, you can find its proton NMR or its IR spectrum and so on to compare it to. Now, sometimes you can't find the things you're looking for in SDBS, so there are other resources too. Sigma Aldrich is a chemical company that a lot of schools will buy their compounds from. And if you type in the name in the search box of whatever compound you're looking for and go to the page for that compound, you can look and see whether they have a link to an NMR spectrum, an IR spectrum, or so on. And if you click on that link, you'll bring up a spectrum here. They've got this is a carbon spectrum in the top line here with the axis numbered on the top, and a proton spectrum on the bottom with the axis numbered on the bottom. And there's the integral curve jumping up over each peak in the spectrum, so you know how many hydrogens each peak corresponds to, okay? Sometimes the trick with Sigma Aldrich is sometimes they have like different grades of this compound. Like maybe they've got a 98% purity and a 99% purity and a 99.5% purity. And often they'll only have the NMR spectrum attached to one of those different grades of product available. So it can be frustrating to, you have to click through a number of them before you finally see an NMR spectrum tab that you can click on and get the spectrum for. By the way, usually it's a PDF file, so it'll download as a PDF file, and then you've got to open that up. Okay, once you've got all of those things done, there's a discussion section. And the previous parts of your report are actually designed to get you to the point where the discussion section is pretty easy, because it's just asking you to discuss some key features. Now, you've already identified those key features in the IR table or the proton NMR table or whatever table you've been asked to get ready. And so it's each of those, in this case, is worth 2 points. You know, I look at that 2 points and I'm going to automatically make sure that I'm talking about at least 2 different things from each spectrum. You know, are there 2 different IR peaks that I think I can identify that make this compound different than all the other possibilities? Is there a carbon NMR peak that I think makes this compound unique? Maybe tells me something that the IR spectrum didn't. Is there an NMR peak that I think is unique about this compound that makes it stand out? And try to find 2 of them in each case, all right? Once you've done that, you've really put together some evidence about why you think this compound is what you say it is. So I mentioned reference citations earlier, and we looked at a table where there was a little superscript 1 at the end of the heading. And that superscript would correspond to a number in a list of reference citations. So here it would correspond to this first one because it was number 1. And so this is that article with the tables of solvent impurities. So maybe we use that to identify the ethyl acetate and the TVME in that NMR spectrum. Okay, and you'll see there are different types of resources here, websites, textbooks, and so on. And you'll see the sort of formatting we expect on these different sources. The format's a little bit different in chemistry than it is in other fields. And you'll be expected to follow this format, but you can see here, for instance, in a journal article, the author's names are listed, then the title of the journal that the article came from, the year, the volume, and the pages. It's not terribly different, but just different enough that you want to pay attention to that. Now, when you get to the end of all these questions, you'll see that the latest version of your answers was saved at some period of time, and don't hit that submit button until you're sure you've finished everything, because you only get 1 chance here, and you want to make sure you've answered everything that you can answer before you click that submit button. Okay, and then you're done. That's all there is to it.